Welcome everybody to is this our penultimate? I think this is our penultimate journal club of the semester. And today we have Jacopo Ferruzzi, who is an assistant professor at the University of Texas at Dallas, where he started, I think, from what I gather, a couple of semesters ago, so August during the pandemic. And he will be, before that, he was in Boston University as a postdoc, and before that, he obtained his PhD at Yale University. And before that, he was in Italy. So he will be talking to us about compressive remodeling alters fluid transport properties in collagen networks, implications for tumor growth. And this is just one paper out of all the impressive work that he has done recently, but we definitely look forward to this specific paper. So please, Jacopo, take it away, and thank you for participating in our journal club.
Yeah, great. Well, thank you for the great introduction, Adrian. And thanks to Emma, Manuel, Adrian, and Johannes for organizing this wonderful journal club. I mean, I really enjoyed the outstanding work that was presented in the past few months. And I'm really happy to be here presenting my own work today. So as Adrian said, I recently started my own lab at the University of Texas here at Dallas. And this paper, the paper that I will be presenting today, is part of my postdoc work, which was carried out, as Adrian said, at Boston University in close collaboration with Harvard School of Public Health. And so that said, I think that I'm the only speaker that has or will touch on the topic of cancer this semester. So I'll start with a brief introduction on why mechanics is even important or matters to understand cancer progression, and in particular in breast cancer, which is quite deadly disease that affects millions of women worldwide, and in particular African-American and Asian women. So traditionally, cancer has been considered a disease driven by genetic mutations. However, the role that is played by the tumor microenvironment in cancer progression is being increasingly appreciated. So as a point of departure, I'd like to just start this talk by summarizing some of the key steps in breast cancer evolution. So the normal breast tissue, shown in this schematic, the mammary gland is a branch structure which is composed by lobules and ducts, as shown here in blue. And this epithelial structure is actually very well organized. And so if we zoom in, we see that luminal cells, here indicated as blue, encircle the lumen of the mammary duct and have the function of secreting milk during lactation. Now, outside of the luminal cell, there are basal or a layer of basal or myepithelial cells, which sit on a basement membrane shown here in purple, which is made primarily of collagen 4 and laminin. Now, surrounding the basement membrane is the mammary fat pad, which is rich in extracellular matrix protein, and in particular, collagen 1. So now, during normal physiology, cells pull on one another, and the generation of the normal mammary tissue structure and function through generation of cell-cell forces is traditionally known as tensional homeostasis. Now, in breast cancer, tensional homeostasis is compromised by uncontrolled growth. So this is driven by genomic instability. And as you can see, epithelial cell will start proliferating in an uncontrolled fashion, will fill in the lumen, and will rupture or break the basement membrane, at which point they can start interacting with the surrounding extracellular matrix. And so the tumor will grow and expand into the mammary fat pad, and they'll initiate steps of invasion. So while we know that initiation is driven by this genomic instability, tumor progression depends on this complex interaction between the cells and the surrounding extracellular matrix, or ECM. Now, just to summarize quickly what is known about the roles that the ECM plays in breast cancer, I would like to highlight a few factors. First of all, mammographic density is a known risk factor of cancer occurrence. So in a mammogram, here you can see a comparison between a low density tissue and a high density tissue, and the mammographic density is known to reflect changes in content of collagen. So clearly, it's a risk factor. Now, ECM stiffening actually plays very complex roles. So there are several studies in which people have mapped the material properties, here, for instance, using indentation tests of biopsy tissues, where you can actually probe the properties of adjacent normal tissues and cells and of the tissue at the invasive edge. And so consistently, you see that the invasive front is characterized by higher stiffnesses. But also in vitro studies show that stiffening of a 2D substrate will induce malignant phenotypes, even in benign cells. So overall, clearly stiffness is somewhat involved in the process of invasion. And then finally, ECM alignment. It has been known to actually facilitate this process of invasion. And in a few seminal studies, it's been shown that particularly radial alignment, when collagen fibers are aligned radially with respect to the surface of the tumor, this is a prognostic marker that is associated with poor survival and disease recurrence. Therefore, it is quite clear that mechanical tumor matrix interactions are actually quite important in the process of initiation and progression of cancer. Now, in terms of the structure of a tumor and the mechanics that changes within tumor, we know that tumor tissues are actually highly heterogeneous. As I said, not only genetically, but also from a mechanical standpoint. And tumor and stromal cells actively remodel the ECM via biomechanical, but also biochemical interactions. And so here you can see actually a multifold image of a tumor where two regions are highlighted, the internal region and more kind of like the boundary with the extracellular matrix. And I would like to highlight some of the known changes that occur in this region, starting from the tumor core. In the tumor core, we have densely packed cells, and in the interstitial space between the densely packed tumor cells, we have elevated compressive stresses due to the deposition of both collagen, but also hyaluronic acid. And this process of depositing extracellular matrix in cancer is actually known as dysplasia. But also there is an increased interstitial fluid pressure. And this is primarily due to the fact that the vasculature in the tumor is leaky and dysfunctional, but also the lymphatics that are supposed to drain the interstitial fluid are also dysfunctional. And so this leads to an accumulation of fluid that therefore increases the interstitial pressure and makes it even hard to deliver chemotherapeutics and nutrients. This as opposed to the tumor periphery or the stromal interface, where it is quite well known that, especially in the early stages or in benign tumors, you have a collagen capsule here shown as this red layer that accumulates as the tumor grows and compresses the surrounding ECM. Now, the early biomechanical changes that occur in this peritumoral ECM during the initial periods of avascular growth are still actually quite poorly understood. And when I started this project, I noticed that a lot of experimental work focused on the interstitial space, and not a whole lot of experimental work was looking at this tumor periphery, the tumor stromal remodeling. And so there were a couple of computational studies, but the idea was trying to get a better understanding and try to fill in this gap. And so the goal of this particular study was to characterize the structural and functional changes that are associated with compression of peritumoral collagen. And so the way we went about this is trying to reproduce these features using an in vitro system. And as a 3D model system, I've been using the multicellular spheroid. And here I'm going to briefly walk you through of how they're formed and what actually I would do in these experiments. We usually culture cells in low adhesion substrate. So after plating cells, these cells, they do not have a chance to adhere to the substrate. So we'll start clumping into three-dimensional spherical structure, as you can see in this time-lapse movie. So here I'm showing an MCF10A spheroid, which is formed from a normal epithelial cell line that is derived from a mammary tumor. And so you can see that after about 48 hours, we form the spheroid, at which point we can actually take it with a pipette tip and drop it into a network of collagen that will polymerize all around the spheroid. And then over the course of the next 24 to 48 hours, we can examine spheroid growth and actually also how it invades within the surrounding extracellular environment. At the end, also something that I have used in part in this study, but also in other studies, is combining optical clearing and multiphoton microscopy, which allows to track the spatially heterogeneous structural properties of the spheroids. Here in this image on the bottom right corner, you can see a near equatorial cross-section where cell nuclei are stained with DAPI, whereas the surrounding extracellular collagen is shown from the second harmonic generation signal, or SAG. So as I said, in this paper, we primarily work with MCF10A cells. These cells generally are considered as normal epithelial cells, but they are actually derived from a human mammary tumor. So when they're implanted or injected in mice, they do not form tumors, but they do express abnormal protein expression profiles and are invasive when cultured in collagen and stiff ECMs. So for this reason, here, MCF10A spheroids were chosen to represent the model for an early mammary tumor. So as the MCF10A spheroid grows in collagen, as you can see in this movie, one can clearly see visible deformation in the surrounding collagen, which here I indicate by arrowheads. If I repeat these experiments in matrices of different collagen concentration, for instance, here 2 milligram per ml versus 4 milligram per ml, we see that despite differences in ligand density, doubling the collagen concentration does not significantly impact the time course of radial expansion, which suggests that the spheroid growth is primarily driven by proliferation. Now, I can treat the spheroid as a growing spherical inclusion, and so one can infer the deformations, which can be quantified as the stretch ratios, radial and circumferential. And we can see that deformations are radially compressive and tangentially tensile in both extracellular environments. And this is just as a sanity check, just to confirm that this is exactly what has been reported in real tumors. In addition, one can look at multiphoton images, and we can see that the deformations that are induced into the matrix by the growing spheroid do not propagate smoothly into the bulk of the collagen matrix, but seem to localize right at the edge, right around the spheroid edge. You can see that if we plot a radial along this dashed yellow line, we plot a radial profile of the SAG intensity signal, and we can see that a locally dense, tangentially oriented layer of collagen reveals itself as a prominent peak in these profiles of SAG signal intensity. So overall, these results suggested that this observed remodeling phenotype could be due to the unique mechanical properties of collagen fibers in response to compressive forces generated by tumor spheroid growth. Now, clearly, that's not the only, that doesn't necessarily mean that that's the only source of this remodeling phenotype. For instance, you can imagine that cells could be secreting their own ECM to try to reproduce the basement membrane. And so the way I went about testing this is trying to reproduce this phenotype into acellular networks. So where you don't have actually cells being remodeling the matrix. And this sort of launched me and my collaborators into this investigation in the properties of collagen fiber networks upon compression. And so just sort of as an intro to the next part of the study, we know that contrarily to other hydrogel materials such as agarose, collagen-1 is actually a physiologically relevant material that is characterized by very complex mechanical properties. And so by understanding more the complex mechanics of collagen networks, it is quite key to increase our current understanding of ECM remodeling in cancer. Just to name a few of key features of collagen mechanics, we can name material nonlinearity upon large deformations, strain-induced anisotropy due to preferential fiber alignment, compressibility, which is loss of volume, and time-dependent responses due to both visco and photoelastic effects. And finally, plastic deformation, also known as inelasticity. And just to sort of give you an idea of how all these properties are somewhat related to interesting biological phenomena, here I'm showing you a high-resolution image of two cancer cells. Those are highly invasive contractile breast cancer cells of MDA and B231 line. And as you can see, these cells remodel the matrix by generating fiber bundles in the direction that separates the two cells. And so you can see that tensile and compressive forces sort of compact the matrix in a way that cells are able to communicate with each other. So there are a variety of methods to probe collagen mechanics. And here I'm just graphically summarizing some of the most popular ones. And starting from the left, you can actually test individual collagen fibrils using an AFM tips or a similar tool. You can perform microbiological tests by employing micron-sized beads, which can be either pulled or twisted. And then there are a variety of micromechanical techniques, including shear rheometry, compression, and uniaxial biaxial tension. And among this last group, shear rheometry is by far the most common testing technique, and data usually analyzed using the theory of linear viscoelasticity. But just so that we keep things in context, my goal here was twofold. First of all, try to induce this compaction in a cellular collagen so that I could reproduce the patterns that were observed in my spheroid experiments. And then second, model the collagen as a nonlinear photoelastic material, because again, these gels are for 80% made of water. And so for this reason, I chose confined compression as my technique of choice. And so in order to perform confined compression tests on collagen hydrogel, I sort of had to figure out a way that this could actually be doable. Confined compression was traditionally developed to test cartilage mechanics. And so there is a very rich literature on that. Try to perform tests in a way that was actually feasible, because these collagen gels are very difficult to handle. I developed a custom setup that basically employs a rheometer. Here you can see in the background, and I equipped the head of the rheometer with the porous indenter. The pores of this specific sinter steel mesh range in the order of 4 to 100 microns, so they are generally higher than the pores of the collagen matrix that we mean to test. And we were able to basically confine the collagen gel using a PDMS well that was basically carved within a 35 millimeter petri dish. So using a sintering piece, we were able to align our sample with the rheometer head. And just to show you a schematic of the resulting setup, here you see my 35 millimeter petri dish with the cylindrical PDMS well in the center, the collagen network here within the PDMS well. We have a glass cover slip, which allows us to perform high resolution imaging. And with the porous indenters, we can apply control strains, which allow us to compact the matrix, but also the porous indenter will then generate an interstitial pressure, which then over time will decay because of fluid flow through the porous indenter. And so in this setup, we regard the z-axis as the loading axis, so you have the vertical axis. And so we can calculate the stretch ratio as one would normally do, and calculate very simply the Cauchy stress by normalizing the measured force by the area of the porous indenter. Now, in terms of data analysis, we can then, and I will mention this a little more in detail in a slide or two, we can actually decouple this the stress at the indenter surface as a stress due to the solid matrix, as opposed to the stress due to pressurization of the interstitial fluid. And so, equipped with this setup, we can now perform a variety of tests. We can perform, as shown on the left, continuous ramp loading, whereby increasing the compression rate, we measure higher forces, again, consistent with pressurization of the interstitial fluid. But also we can perform step loading. So we can apply compression and then hold the indenter at that height. And where then we'll see a decay of the measured forces, which I guess initially surprisingly, but not so surprisingly after all, will basically decay almost to zero as the interstitial fluid flows through the matrix and the force decays. Now, this type of deformation can be applied also by using multiple steps, not only one step. And so here you can see transient responses on 5 compression steps. And then we can use the steady state force level to actually characterize the material properties. So we can normalize, as I said, the steady state force by the area of the indenter and plot that stress versus the compressive stretch. And so here you can actually get an insight of the material properties. And so one thing that really jumped, maybe biased by the literature on cartilage mechanics, one thing that really jumped immediately to my eye was the fact that these networks are extremely soft. As you can see here, the force decays almost always to zero, so you don't see that nice step-by-step increase in force. And as you can see here, the stresses that are developed within the network are actually quite modest.
Jacopo, so, can I interrupt real quick?
Yeah.
Sorry, I'm just extremely curious because we've tried to do similar experiments and we had so much trouble with essentially what you're describing, right? That in these gels, the forces tend to go to zero very rapidly. And then there was always a challenge for us that the forces of the interaction between the porous indenter and the wall, those frictional forces, were in the same order of magnitude as those equilibrium forces we would measure. And if we reduce the diameter of the indenter to avoid those frictional forces, we would have significant leakage through the sides. So sort of the assumptions that we made when modeling these problems were pretty severely violated. Did you have those same issues? We did not use PDMS, so we used rigid walls. So I forget, some sort of polymer. So I was wondering whether you faced those same challenges and how you resolved them.
Well, I did indeed face the same challenges. I mean, that is generally something that was not only, is not part of what you are referring to, does not only apply to hydrogels, but generally when performing this test, right? Theoretically, you should have the diameter of the porous indenter is exactly the same diameter of the constraining well, right? So I even found differences if I constrain my material with PDMS, or for instance, right now we developed a machine sinter-sealed confinement chamber. We have tried, ultimately the way I resolved this is by having an indenter, which is about 8 millimeters, and having the PDMS well being 9 millimeter. That is, there are 500 microns on either side. I reduced that distance or that gap to 250 on either sides, and we saw no significant difference in the results. We would just have, because again, this is not perfectly aligned all the times, right? So from time to time in the experiments, you would hit the PDMS well, and so you clearly see a much higher jump in force, so we'll just discard those samples. But in this context, the 9 millimeter well versus 8 millimeter indenter is what worked for us. And we didn't see a significant effect by decreasing it to 8.5 millimeters.
Okay.
And it's something that, even in the literature on cartilage mechanics, there is always a little bit of gap that is left on the side.
Appreciate it. Thank you.
Yeah. And so even, I guess that Manuel Commons arrived at a great time, because again, this was a frustrating point in this investigation, right? We would see this type of fairly low forces, and so we sort of from, again, coming from a background where we always want to try to pair the structural properties with the mechanical properties, I started wondering what I could do to try to titrate both the transient and material responses. And so in terms of microstructural properties, two straightforward things that one can do to modulate the properties of the collagen networks is by varying the density of collagen. And in this type of in vitro culture studies, it's common to vary the collagen concentration between 1 and 4 milligram per ml, and then try to tinker with the cross-linking. So there are a variety of ways of cross-linking collagen matrices. Here, I just used a standard technique, which is by using glutaraldehyde. Preliminary tests suggested that 0.2% glutaraldehyde are sufficient to increase significantly the material properties of our hydrogels. And so you can imagine here in this little cartoon, I'm sort of giving you the way I imagine the networks after cross-linking. Glutaraldehyde is a bifunctional molecule, so it will increase the strength of intrafibrillar cross-link, but also interfibrillar cross-links. And so, so once we perform those structural modifications, I looked at the resulting microstructure using multiphoton microscopy. And so here you can see second harmonic generation images from left to right of 1 through 4 milligram per ml gels, and the control gels are shown at the top, and the cross-link gels are shown at the bottom. And this really shows that, qualitatively, that cross-linking, because it was actually done post-polymerization, does not really impact qualitatively the structure, but also quantitatively, we quantified fiber structure using an open source software called CityFire for segmentation of collagen fibers. And here I'm showing that fiber density really depends on the collagen concentration and is not really impacted much by the degree of cross-linking. But now just wondering if what would change, at least if we can get a readout of the changes that occur with cross-linking. We found that as a functional counterpart to the second harmonic generation signal, we can actually use 2-photon fluorescence signal here shown in red. And we saw that cross-link network exhibits significantly higher autofluorescence at all concentrations. And this indicates that treatment with glutaraldehyde alters somewhat the physical properties of these individual fibers. And so after this microstructural characterization, what I did is started investigating the effect of cross-linking on the confined compression responses. And so one of the first experiments that we did is imposing compression steps up to 80%, up to 80%, so severely compacting the matrix. And we found that controlled gels display very high force peaks and a smooth plateau of stresses, which is indicative of plastic deformation. On the contrary, cross-link networks display lower peak forces. So you see that the peaks are always consistently lower than in the blue curves. And actually you see a marked stiffening, which then leads to sudden rupture above 20% strains. So this sort of was an initial test to sort of suggest that if we're interested in the elastic properties, we should keep our measurements below 20% compression. And that's what we did moving forward. So we performed subsequent tests up to 80% compression. And by superimposing the material behaviors, it's quite clear that cross-linking induces material stiffening. And particularly, this stiffening is quite nonlinear. And in terms of trying to understand if there is a change in energy dissipation, what we did is we used a generalized Maxwell model that is a linear combination of an infinity of springs and dashpot arranged in parallel, so that we could actually extract the relaxation time spectrum at each step of compression. So the way we obtain those spectra is by using this type of model, which again is a generalization of the Maxwell model, where we do not constrain the amount of time constant, the number of time constants that are involved in the stress decays. And this allows us to back solve for this continuous relaxation time distribution function. And so we actually see that the spectra for collagen usually consist in 3 to 4 distinct peaks, and the heights of the peaks increases with compressive strains, and is always lower for cross-linked gels. So as you can see here, the red peaks are lower than the blue peaks, which are for the control gels. So what we can see is that if we calculate the area under those curves, this gives us a quantification of the amount of energy that is dissipated at each step of compression. And we see that generally the dissipated energy increases with increasing deformation, and is always much higher in control networks than in cross-linked networks. And so overall, this analysis sort of suggests that there is not only one mechanism that is involved in energy dissipation in collagen networks. And so the first clear candidate was poroelasticity, that is looking at the role of interstitial fluid flow. So that's what we did next. So we employed the theory for biphasic mixtures that, again, was developed in the context of cartilage mechanics. I have already mentioned this a few times, so we used that literature as a motivation for this work. And we used this theory to fit data from confined compression of collagen networks. So here we are basically assuming that the bulk mechanical behavior under compression can be separated into a deformation-dependent stress, which is generated by the solid collagenous matrix, here shown here in the bottom, and a transient pressure that is generated by the fluid that is filling the interstitial space, here shown at the top. And in terms of causative assumption, we assume that fluid is treated as inviscid, that is, it supports a pressure and not a shear stress. And furthermore, that the collagen matrices can be characterized by an isotropic hydraulic permeability. This makes sense because microstructurally, you see that these collagen gels do not have any preferential direction. And furthermore, we tried, I'm not showing it here, but as an aside, we tried to impose a deformation-dependent hydraulic permeability. That is assuming that the more we compress, the more basically the lower the permeability becomes. And we did not see any improvement into the quality of our estimations. And so we kept it as simple as possible by maintaining a deformation-independent isotropic permeability, at least for the fluid phase. In terms of the solid phase, we treated the material as hyperelastic, and among the many candidates for strain energy function, we use a Yale material model. And again, the rationale for this is just trying to use the simplest material model that reproduces the nonlinear material behaviors that we observed experimentally. And here in particular, we can see that we decomposed the strain energy function into an isochoric and a volumetric part. And this allowed us to account that for the decreasing mixture volume during confined compression, and we could still enforce incompressibility of the solid phase by imposing that the determinant was J here, the determinant of was equal to 1.
Jacopo, another quick question. I'm surprised you use the U model just because I never see anybody use it. Is there a reason why you didn't use? I naturally would go to the Mooney-Rivlin first, or the Ogden. Was there a reason you didn't use either of them?
Well, I started, I guess, in a traditional fashion. I sort of started with a Newcan and then moving to Mooney-Rivlin forward. Blatzko is just a linear combination of these two, and neither of these models gave us satisfactory fits to the data. So only when moving to this nonlinear form, which is quite phenomenological again. So the goal here is not to try to capture the microstructure. We would do that differently, but the goal is to actually reproduce the data and also keep this strain energy function quite simple. The reason for that is actually the next step. So again, thank you for bringing up that point, because once we take these two causative assumptions and we plug them into our linear momental balance for the mixture, we actually get a governing equation. Again, this is from the linear momentum balance, in which, because of the axis symmetry of both gel geometry and external load, now the displacement field, here indicated as U, is only dependent from the axial position Z and from time. So this gives us sort of a manageable equation to sort of solve the biphasic problem. And so all in all, this results in a nonlinear parabolic partial differential equation, or PDE, that then I went on and solved using combining Newton's method with an implicit finite difference formulation with the appropriate initial and boundary condition. So I hope that that sort of clarified the rationale for using this. And so once we implemented this, you can see I adopted a two-step fitting procedure, again, being inspired by the work of Gerard de Teschen, in which first we fit the material responses to backsolve the material parameters for the Yale model. And then we fix those parameters and we fit the transient responses, the ones that you can see in this slide, to backsolve the hydraulic permeability.
Did you manually fit these, or did you use like a least squares approach?
Least square, yeah. So
why did you split the fitting procedure rather than fitting them simultaneously? I'm just curious, because I always struggle with making that decision.
Well, again, as I said, I don't know if I have a great answer for that. I think that it made a lot of sense, right? Because as I was trying to rationalize these results, you tend to look at the transient responses, so these decays that I'm showing here, but also the material properties, that is the equilibrium responses. So it would make sense to me to fit the equilibrium responses to the strain energy function for the material, fix that, and then just have one free parameter to describe these decays. And again, because those structures are quite porous, we could get away with actually maintaining the hydraulic permeability quite constant, up to 18% strain, which is what we used here. Again, probably I was biased by it because of Gerard de Teschen's work and proposing this two-step procedure, but it ended up working quite well for me.
Did you say 18% or
1/8? 18%. Yeah, it's funny because we have 3 minutes of hold phases, but also 3% strain at each compression. So here you see that in terms of time, we get to 18 minutes, and then gets also 18% strain at the end, which is quite anecdotally a coincidence.
Quick question. So if you had gone to larger strains, do you think that deformation induced anisotropy in the permeability tensor that you ignored for now would have mattered more?
I probably would have, yes, definitely. And this is something, I'm just going to scroll a few slides back. Let's see if this is going to work gracefully. You see, this is what we did here, right? We compressed this, we did extreme compaction of these matrices. And you see that, again, the peaks in force, which again in a photoelastic framework we impute completely to pressurization, become much higher than in the cross-linked networks. And so you have this extreme pressurization that would happen, and as you can see, even materially, the matrix will either plastically deform or collapse. But then when you get to the extreme compaction, then you will also pick up the stresses. So you definitely have this very nonlinear effect, both materially and in terms of permeability. But that's not what we sought to try to recapitulate here. And so we kept it again in what we thought was the linear elastic range, or the elastic range, even if nonlinear.
Thank you, appreciate it.
And so to sort of like summarize these results, here I'm showing bar plots showing the energy storage. This is energy stored at 18% compression, so at the maximum deformation that we imposed. As you can see, the energy storage for control networks, those white bars, it's actually not affected by concentration, a little bit surprisingly. Whereas the primary determinant of energy storage and material properties, therefore, seem to be cross-linking. Here you see that there is quite a distinct step when you go from 2 to 3 milligram per ml, and you can actually see it in these responses here. If you look at the cross-link behavior, you have a quite modest response for 1 and 2 milligram per ml gels, and this actually jumps quite significantly between 3 and 4 milligram per ml. And this is what is captured here in the black bars. And so here I'm just simply showing a plot of the shear modulus for a 3 milligram per representative 3 milligram group. And as you can see that the shear modulus varies non-linearly, and it's quite higher for cross-link networks. So all of this sort of led us to conclude that it's the cross-linking that primarily controls material behavior in these networks, obviously in a concentration-dependent way. And as we move to looking at fluid transport properties, here I'm plotting the hydraulic permeability. And so what you can see is that the permeability decreases with increasing collagen concentration, which again is to be expected because of you're going to have more frictional losses caused by the interstitial fluid that flows through a denser matrix. Again, what we would expect from Darcy's law is what is captured in this plot. What was surprising to me personally at the beginning was that, yeah, okay, density controls fluid transport, but actually we found that control gels at consistently lower permeabilities. And this is something sometimes was significant, sometimes was not, but it was quite consistent in all the groups. And this was surprising because if you recall our structural quantification, we do not see any difference in density between control and cross-link networks. And so this changes in permeability, which then lead to higher pressurization in control networks. It was puzzling to me, and the way I went about this is just by visualizing what actually, if anything, had changed during our compression test. So after compression, we would again perform multiphoton microscopy, now at a less magnified scale. We'll actually try to image the entire thickness of the gel. So here you're looking a lateral cross-section of the gel, where the cover slip here is indicated by the dashed line. And basically the indentation surface is this free surface up here. And so when we compare control and cross-link gels, we always see that control gels are shorter, are more compacted. And here at the interface, they show this densely, this plastically deformed interface that was formed at the surface where the indenter applied deformations. This can be quantified by looking at the thickness of the gels. Again, the white bar are the control gels, whereas the black bar are the cross-link gels. And if I didn't mention it before, these gels are all 3 millimeters at the beginning, so they all start from the same height. But then after deformation, we can see that there are remnant plastic deformation, especially at lower collagen concentrations. And there is a consistent differences between control and cross-link networks. So the cross-link networks behave more elastically. They will recoil back after compression, whereas the most of the control networks, they will basically stay there. They will be basically plastically deformed. And this was actually, to me, quite interesting because it really reminded me of what we had seen as radial trends around our spheroids, our tumor spheroids growing in collagen. So if we now indicate with this zeta coordinate, an axial coordinate, we now can plot the mean intensity of the second harmonic generation signal over the depth. And so we can see that there is this marked peak, again, reminiscent of what we have seen surrounding the collagen, surrounding our tumor spheroids. And so I guess that what we started calling this as this phenotype a compressive remodeling phenotype. It seems that compression is really what is driving this compaction, this localized compaction of collagen, and wonder how can we describe, or what are the mechanisms that are underlying this compressive remodeling phenotype. The continuum model, because it assumes a fine deformation, may not be able to capture this localized deformation that actually are occurring here. So we had to resort to a different tool. And in particular, we resorted to discrete network modeling. What happens if we were inspired by some of the work from the group led by Roger Kam at MIT in modeling cytoskeletal network. Briefly, we basically treated the network as we form networks to try to reproduce what we have measured experimentally. The governing equation is a glorified version of Newton's second law. And we endow the fibers with, each fiber could store energy either by stretching or bending. And in both cases, the key parameters is the elastic modulus of the fiber. Because we segmented the fibers using segments that are smaller than the persistent length, we could treat each segment as a linear elastic spring. And then the additional thing that we did that was, I guess, novel is treat cross-linking the network and treating if two fibers form a crosslink, the node where the fibers are exchanging forces basically is transmitting the forces experienced by both fibers, and they're moving together. And basically what happens here is that we used a breaking force or a threshold force. So when the force exchange to the crosslink will hit this threshold value, then the crosslink will rupture and the two fibers will be free to move individually. And so the first thing that we did is try to reproduce some of the stress relaxation behaviors that we observed experimentally. Those obviously we were trying to qualitatively reproduce this behavior, but this gave us some insight on the behavior of the network. And so in fact, we see that the primary mode of energy storage is bending, and the decays are primarily due to the decaying bending energy, whereas stretching energy, which is 1 order of magnitude lower than the bending energy, it's only accumulated during the step of compression. Another thing that we saw, and this is something that you can see here a little bit in the video, is that you start seeing this compaction of collagen fibers at the boundary that is undergoing confined compression. And we could see that again by plotting, by using this zeta variable, and by plotting the node density before and after compression. So here you can see before compression, the red line has a quite constant node density at all depths, whereas after compression we see this densification. In addition, if we plot both the azimuthal angle and the polar angle, sort of giving us an idea of what are the direction or the orientation of the fibers, we see no different in the azimuthal angle, whereas the polar angle is being completely compacted around 90 degrees, which means the fibers are buckling or they are rearranging under the compressive load, perpendicularly with respect to the direction of compression. Now, this model was informed by a variety of parameters. Part of some of these parameters, we could measure them experimentally. Some of them we had to resort to the literature. But the two key parameters that control the network behavior were identified as the fiber stiffness and the strength of crosslink, or the breaking force between covalent crosslinks. And we have some information of what the fiber stiffness is from the literature, so we could sort of narrow it down into reasonable range. Whereas there is no way of knowing what is the crosslink strength. And so what we did is we varied those parametrically. And sort of in the last thing that we did, we sort of like looked at how parametrically these two factors affect network behavior. And so here from the left, for a pair of values of Young's module and crosslink strength, we see that a network at low fiber stiffness and low crosslink strength, upon two steps of compression, we see a stress behavior that reminds us of the control or non-crosslink network, where the force or the stress decays basically back to zero. Now, what we can do is actually gain some insight from the model, and we can actually see that at each step of compression, crosslinks are broken, so we lose the number of crosslinks, and there is a modest bending energy that is being accumulated in the network as a whole. Once we remove the deformation, this network will recover only to about 30% of its original height. Again, sort of reminding us that this is what this is indicative of what is happening in our experiments. Next, we can actually just increase the Young's modulus while keeping the cross-link strength pretty much the same. And you can see that we don't gain anything in terms of stress evolution. We actually rupture even more cross-links, and that sort of makes sense. You know, we have basically a network formed by more rigid sticks, but the glue between them is still weak, so they are going to be compacted. We have more bending energy, but basically even less elastic recovery. And so it's only by increasing both fiber stiffening and breaking force or crosslink strength is that we see behaviors that are indicative of our crosslink network with virtually no rupture of crosslinks, higher bending energy, and a virtually full elastic recovery of the network. So what this is telling us is that inter and intrafibrillar crosslink play a clear role. If we strengthen them, we can actually reproduce the mechanical responses that we see in chemical crosslink network. And by having weak crosslinks, it is what underlies the plastic remodeling of the collagen networks that we see after compression or around our spheroid. And so just in the interest of time, I'm afraid that I've gone quite long, so I apologize. In the interest of time, I'll summarize what we found is that tumors growth remodel the peritumoral collagen under compression. And we see that this localized densification impairs the fluid transport properties of the surrounding ECM. And overall, this compressive remodeling phenotype is driven by buckling of fibers and rupture of crosslinks. And so now we basically can infer at least that the fluid transport properties of the tumor microenvironment can be altered by ECM compression. And so this implies that interstitial fluid pressurization can also arise from peritumoral ECM remodeling and not only by a leaky tumor vasculature. In terms of limitation and some of the future direction, obviously our collagen hydrogels are the most simplified model of a tumor ECM. So obviously more realistic model, for instance, collagen hyaluronan co-gels would need to be considered to sort of gain more of an insight of what happened in tumor ECM. I guess one key question is whether or not this remodeling phenotype plays a significant role in the malignant transformation of benign tumors. It is known that compressive forces can make cancer cells more migratory. It is still an open question whether this fibrous capsule that is generated around the tumor actually plays a role in that process. And in addition, what we did here is sort of use macroscopic mechanical testing to get an insight of the mechanism driving this remodeling phenotype. But as I said at the beginning, this tumor matrix interaction are actually quite spatially heterogeneous, and so we would need mechanical probes that are local and also appropriate models to actually process this data. And to sort of highlight why there is the need for that, here I'm showing you two tumor spheroids embedded in collagen. On the left-hand side, the MCF10A spheroid that induces this compressive remodeling phenotype that basically was the focus of this study. But also you see that more contractile, more mesenchymal-like cells, cancer cells, induce a tensile remodeling. So that then aligns the collagen fibers radially and then leads to very active invasion. And so I'm sort of leaving this with one of the movies that you can actually acquire by comparing these two remodeling phenotypes, again, highlighting the need to have to be able to describe these processes in more details in terms of tumor matrix interactions. And so with that said, I'd like to acknowledge my previous groups at Boston University. As I said, this was part of a larger project with our THN of School of Public Health. I'd be remiss if I didn't acknowledge the students that helped me co-author this paper. Meng San was essential in developing with me the fiber network model. Anaita Pilvar, who ran the multiphoton imaging studies, and Anastasia from the Catherine Zhang lab, who helped me with the analysis of the continuous relaxation time spectrum. and obviously funding agency from both NIH and the Department of Defense funding this project. And as in the introduction, ever since we completed this works, I moved here at UT Dallas. So if you are interested in discussing about this work or any other work, I would be really happy to hear from you. And thank you very much for your attention.
Thank you, Jacopo. That was a lot of information, but very well presented. Very, very clear. So yeah, thank you very much for giving us all the details, all the scoops. Questions?
I thought that was the goal, right?
Yeah, no, I actually really enjoy getting all the in-depth of all the methods, in particular, all the methods and the results. So yeah, very, very nice. I also really like, some experiments, some computation, but that's my bias. But questions for Jacopo? Maybe we have time for a couple of questions. Anyone wants to jump first?
I'm quite curious, Jacopo, what are you going to continue doing now as a PI? You seem to indicate that you're still doing the confined compression test. So I'd be curious for you to tell us a little bit. What are your plans? How does this integrate into your future work?
Well, I mean, I think that was sort of like my first, the way I sort of launched into this area, which really, as you can see, this study was a, we used these phenotypes around our tumor spheroid to sort of launch into an investigation on collagen network mechanics. So as I sort of left you at the end, we see even more complex and more, I guess, pathologically meaningful phenotypes that still deserve investigation. So this is something that we are trying to see if we can actually make a nice contribution with. In terms of confined compression, as you were asking, that's now a tool in the lab, right? That's something that we're using with some of our collaborators to look into the properties of some excised tissues, not necessarily collagen or fibrin gels, which is, I think, what you'd be probably most interested in.
Other questions for Jacopo?
By the way, one thing that I really liked was your analytical or semi-analytical solution to this pretty complex problem. Seemed like you had learned something from Jay in terms of simplifying a geometry and,
well, that's
saving yourself the pains of solving a full finite element problem.
Well, I mean, we can do that, and I think that my grad student is definitely here in the audience, and we are using FEBio also to simulate some of those systems. But as you said, I learned from Jay the way, and I'm trying to pass it on down to my grad students. Let's first get an understanding ourselves, and then obviously as a sanity check, you always should match up with our FEBio models. And we can, obviously, as you commented, I used a fairly simple constitutive model so that this was actually doable. And so for more structure-based models, we will have to resort to the finite element models. But having an understanding of how the semi-analytical solution works, it would really guide our future work.
Yeah, look, I really like that. I think and maybe my group, we can definitely learn from that. We tend to start with the most complex models and then, struggle with that. So I really appreciate that.
Any other questions for Jack? Well, maybe one last question, and then we'll stop. Probably people have things scheduled maybe for.
Well, I can ask one last question. Sorry, Vivek.
Okay.
I'll let you go ahead. Sorry, I thought nobody was going to.
Okay, yeah, sure. So I had a question about the discrete fiber network models that you're using. So for a full-scale boundary value problem, you would want to consider upscaling this model, right, to a continuum model through some kind of homogenization. So have you considered any study of that kind for this particular model?
Well, let me see if I understood your question. You were asking about the fiber network model, and if we were to use a continuum model, we did indeed use a continuum model. That's what Manuel were discussing about. But obviously, because we see, a limitation of the continuous model is this assumption of a fine deformation, right? That the fibers will move according to the microscopic deformation. And that clearly, after we performed the continuum modeling, we did the imaging after compression, we saw that that was not necessarily the case. And so we had to sort of like go through the bother of developing this network model to sort of like capture the localized densification and tangential alignment. And so that was sort of like, we basically took both routes, right? And we learned different things from both approaches. So that generally, if anything should come out of this, is that you should always, depending on what the question that is being asked, you may want to consider different types of models to try to separate different effects, which was indeed the case because the sort of the poroelastic effects were highlighted by the continuum model through homogenization, whereas more this viscoplastic and non-affine effects were highlighted by the network model. So we learn different things from the different approaches.
Okay, got it. Thanks.
Cool. Well, I think I will probably say we stop right there in case people have other meetings that they have to attend to. But thanks again, Jacopo, for the great presentation and for all the details. Really appreciate. I'm trying to find the. Oh, there we go. I'm trying to find the clap on the reactions. So